Alright guys, so we are going to look at something called mitosis monster, and we're going to discuss mitosis, and they're going to basically go through the steps of mitosis on their desk with some string and these fake chromosomes that we have. It's really just toothpicks. So I'm going to go walk through this with you. You guys, what you're going to be graded on is basically that you have copied in and completed these notes. I know, like, if you have string and stuff at home that you want to use, that's fine, but I'm going to walk you through what we do in class. So it says we have an organism that normally only has 6 chromosomes in their body cells. Now this is where I would ask your classmates, how many chromosomes do we have in our cells? We've not talked about it in here, but it's hopefully something you've heard at some point in time in a science class. And usually at least one person knows that we have, I'll hear the number 23 or 46. And we have 23 pairs, which would give us 46 altogether. So, right, we have like chromosome number 1, for instance. We got one from mom and we got one from dad. So there's our pair. And we have all 23 chromosomes. We have one from mom, one from dad. So we have 46. Now this organism though, not everything has 46 chromosomes, and I have a list of things that I'll usually read off, and like here, let me see if I can look it up real quick here. Numbers of chromosomes in different organisms. All right, so if we're just looking at a few things here, even this one, humans have 46. Let's see if, oh, it's not gonna let me go to it. Trying to find one that I can easily read. Well, even here, like if you look at a roundworm, like a roundworm is like dogs can have roundworms. What's make their belly? It's why we worm them when they're little puppies. They have 4 chromosomes. Rice has 24 chromosomes. Humans have 46. Chimpanzees have 48. Dogs have 78. Now, it does not mean that they have more genetic material. Like, their chromosomes might be a little smaller. I don't know off the top of my head, but they could be a little bit smaller. Let's see what else we have here. Maybe here. A pea plant has 14 chromosomes. A frog has 26. A chicken has 78. So it's got a lot more than we do. So anyways, that's just some different numbers here. Buffalo have 60, cat 38, cattle 60. So just so you know, like every organism has different numbers of chromosomes. Let's see. There we go. Sorry, right here. All right, so anyways, our monster happens to have 6, and I just picked a nice, easy, small number so that we weren't counting out chromosomes through the entire class. All right, so the organism got a haircut, and now the hair must grow back. So we're going to have to think about our cell process and how it's going to be our ribosomes in the cell that have to make this protein to grow this hair back. The problem is okay, well, how do we make this protein? How do we make new cells? So we have to make new cells. So how does this happen, though? So the first thing I'm going to tell them to do on their desk is make your pretend cell with 6 chromosomes in the nucleus on your desk. So what they're going to do is they're going to have this string, and they're going to put it into a circle. And then they're going to have another string that's red. That's going to be our nucleus. And from there, I'm going to tell them to put 6 chromosomes in there. Now they have little broken pieces of toothpick. And so I'm going to have them put 6 of these toothpicks in their nucleus, because that's where our DNA is, right? It's where all of our information's at. So they're going to they're going to build this on their desk with their string and their toothpicks. All right, so if we have to duplicate this cell, what is the first thing that needs to be done and why? So I'm going to tell them, like, hey, we have this on our desk right now. What we need to have on our desk by the end, we're making a brand new cell, right? So I need 2 of these cells. Sorry, guys, this is not pretty. But I need 2 of these cells. So how do I go from 1 and 2? Well, it's going to split eventually, but I can't just split this. If I were to try to split this cell in half right now, look, if I just split it in half, that's not what I need. I would have a cell with 3 chromosomes, and I would have a cell with 3 chromosomes. Now, if these cells split, I would have 1 1/2 and 1 1/2, and I can't do that. That can't happen, right? Like, I can't have half the number of chromosomes and keep halving it. So the first thing that needs to be done, and usually I'll try to pull this out of your classmates, is we need to double our chromosomes. So right now in your body, you have cells in your body right now that are making new sets of chromosomes so that this can happen. Because everybody right now is making new cells. You guys are losing skin cells like crazy every day. So we're constantly having to make more, right? Like if we didn't make more skin cells, your skin would wear down and we'd eventually see muscle and bone. So we need to double our chromosomes. That is the first thing, and that is so that our new cells can have the correct number of chromosomes, right? So that's the first thing, always first thing, no matter what. If we're making new cells, the first thing that has to happen is doubling the chromosomes. So what I would tell them to do right now on their desk is I would say, okay, get 6 more chromosomes or toothpicks, get them and stick them in their cells. So at this point, they now have 12 inside this red nucleus. All right, so once we finish the step above, what has to happen before the cell can split into 2 separate cells? So right now what they have, let's see here if I can get my drawing here. I'm gonna add my chromosomes, 1, 2, 3. So these are the new ones that they've added. So right now they have 12 chromosomes all just floating around in here. So what they need to do is they need to split these chromosomes up. All right, so before the cell can split into 2, we need to split our chromosomes. Okay, so we're going to split these chromosomes up. So that's what we're going to walk through here. So on their desk, they would be doing this and physically with their hands building a new cell with these strings and these toothpicks. All right, so we're going to look at the different phases here. So what happens is, and to split our chromosomes up, I'm going to write this here too, we have to break down that nucleus. So we have to break down the nucleus, right? If we don't break down this nucleus, we can't split these chromosomes up. So what happens is, go to drawing. I'm going to do it in red. I'm doing it in red because their string is red. So their nucleus starts to break down, and we start to see, instead of this nice thick red line like we had up here, we start to see it breaking apart. You can start to see chromosomes. 1, 2, 5, 6. 1, 2, 3, 4, 5, 6. Sorry, I'm just trying to make sure I have my 12 chromosomes. So this is what they have right now. And this is called prophase. You'll hear me say PMAT a lot. PMAT, P-M-A-T, prophase, metaphase, anaphase, and telophase. So after prophase, when this nuclear membrane starts to break down, we're going to see where it is completely, like this in metaphase, it's completely gone. It's completely gone, and all my little chromosomes here have partnered up. And a lot of times you'll see them as X's like this. So I'm going to go ahead and I'm going to, well, I thought I was going to erase, here's my eraser. Okay, I'm going to go ahead and I'm going to erase these. And I'm going to make them as an X. So what you have here is you have, I know they look like an X, but really one chromosome, I'm going to kind of show you here. One chromosome is like this. The other chromosome is like this. And they're attached in the middle, right here in the middle. So like I'm going to do a little dot in the middle of these. They're attached right here in the middle. Okay, so what this is called, this phase is called metaphase. And I always think of meta, M, middle. So meta, middle. So they've lined up down the middle. Now what we also have here, let's do some more amazing drawings here. See if I can do this. They look like little tin cans. And I call these my team captains. They're called centrioles. I'll label them. I'm going to have you guys label yours also. Centrioles, and these centrioles have spindle fibers that come out of them. And what they're doing is they're attaching to the middle, and they're going to pull these chromosomes apart. All right, I'm not going to draw all of them. So this is metaphase. I'm going to make this larger here. Let's make this one larger too. All right, and then I'm going to label this. This is centrioles. And that's those guys right there, centrioles. I have spindle fibers. And that is the stringy things coming off. Now, as far, I'm going to draw a large, kind of a large X here. This right here in the middle, right there where these two things are attached, it's a little bit smaller, is called a centromere. Okay, that's what this thing in the middle is called, is a centromere. Now each one of these are called, there's one chromatid, and here's the other chromatid. Do my dot in the middle again. Okay, so my centromere is that dot. Each one of those thick lines that X that part and that part, that is called a sister chromatid. Trying to find somewhere to put this. A chromatid. Now it's your DNA. They call it chromatid, they can call it chromatin, a chromosome. It's your DNA. Okay, that's your DNA there. Okay, so the chromatid is right there and also right there. All right, so I always take metaphase and I label everything on metaphase. So I said P mat. So my next one is A, or what you guys would know as anaphase. And this is where M and M, meta, middle, ana, apart. So I'm going to make this a little bit thicker. But what I would have the kids on their desk right now is start to pull their chromosomes apart. I'm running out of room here. Now I'm not going to draw the centrioles and the spindle fibers, but they are still there. I'll draw one right here. They're still here and they're still pulling these things to the side. Now what happens is sometimes they don't pull apart like they should. And we'll learn about that. Like what happens if this one doesn't pull apart and it's still attached over here? You can get things like Down syndrome because of that. All right, so anaphase, apart, it's pulling apart. And then P-mat, the T is telophase, and I think of two, tele2. So what you're going to get is you're going to start getting this cell that is going to split into two. So you get kind of this, and sometimes they'll say that you're going through cytokinesis, which is the splitting of cytoplasm, and that's what is happening right here. But in telophase, you start to get your nucleus forming back. And then your chromosomes, they unwind and they uncoil. And when they do that, it just makes them look like there's a mess of chromatin. This is called chromatin. So chromatin is just when it's a dense mass in there. When they coil up and you can actually see them, like in metaphase and anaphase, it's called chromatids. chromatid. When it uncoils and it just looks like a mass of junk, it's called chromatin. So I'm going to go ahead and I'll write that out to the side here. Chromatin. Once again, it's the same thing. It is your DNA. That's all you need to really know, is that's your DNA. All right, so that is PMAT. That is mitosis. Mitosis is not the division of a cell. It's not cell division. If you notice, everything we're doing is we're doing to this nucleus. We're breaking the nucleus down. We're taking the chromosomes, we're lining them up in the middle, we're splitting them, and then we're reforming the nucleus. So this is the division of the nucleus. That's what mitosis is. A lot of people think it's dividing cells. It's not. It's division of the nucleus. So where does a cell spend most of its time? Well, in the cell cycle, mitosis, or PMAT, is just a short portion of that. Most of the time, the cell is spent in something called interphase. Now, interphase is where most of your cells are. You have a lot of cells in interphase right now, but you have a ton also going through each of these steps in PMAT. So in interphase, what that looks like is you just have oh my gosh, this mouse, a circular blob, and it just looks like a big dark circle, and it's your nucleus, because your chromatin at the moment are unwound. Now during interphase, you're making new chromatin. Your cell is growing, your cell is getting ready for mitosis, and most of your cells are in interphase. All right, so let's look at what happened here. At the beginning of mitosis, we started with a cell that contained, so at the very, very beginning, guys, you had 6 chromosomes. All right, so we had 6 chromosomes. At the end of mitosis, we had 2 cells. We're forming 2 cells here that each had 6 chromosomes. That each had 6 chromosomes. Now, what I did not put in here is the middle part, that we went through interphase, because interphase is where we are copying. I'll put it here. The big thing here is we're copying chromos. So during interphase, we would copy and we would have 1 cell with 12 chromosomes. At the end of everything, we end up with 2 cells that each have 6. All right, so what I have them do is they built this on their desk, they've done all of that, and then they're filling this out, and I have them right here. Here we go. 6 chromosomes. And then I have doubled to 12. I'll have them put that there. And then we have 2 cells, each having 6 chromosomes. All right, and we'll stop there for that first day. I'm sure we won't have time to start meiosis 1. We'll talk about that in a different video. But right now, I want you guys to fill this out. Your homework is going to be that you show me that you have filled this out and send it back to me completed. All right, if you have any questions, let me know. I'll talk to you guys later.